Introduction. DNA repair is an essential process that maintains the integrity of the genome by correcting various types of lesions and damages to DNA. The accurate and efficient repair of DNA damage is crucial for the survival and normal functioning of cells. Failure to repair DNA damage can result in the accumulation of mutations, leading to genomic instability and the development of various genetic diseases, including cancer. In this script, we will explore the different mechanisms and pathways involved in DNA repair, as well as their implications in genomic stability, genetic diseases, and personalized medicine. Direct repair is the simplest form of DNA repair mechanism, where DNA lesions are directly reversed without the need for any template or involvement of enzymes. One example of direct repair is the repair of methylated cytosine residues by DNA methyltransferases. These enzymes can remove the methyl group and restore the original nucleotide sequence. However, direct repair mechanisms are limited to specific types of DNA damage and may not be sufficient to address more complex lesions. Base excision repair, BER, is a versatile and prevalent DNA repair pathway that removes damaged or inappropriate bases. BER involves a series of sequential steps, starting with the recognition and removal of the damaged base by specific DNA glycosylases. The resulting apyrantic/apyrimidinic AP site is then processed by AP endonucleases, DNA polymerases, and DNA ligases to restore the correct nucleotide sequence. BER primarily deals with small base lesions, such as oxidative damage, deamination, and alkylation. Nucleotide excision repair, NER, is a highly conserved and versatile repair pathway responsible for the removal of bulky DNA lesions, including UV-induced pyrimidine dimers and chemical adducts. NER involves multiple steps, starting with lesion recognition by a complex of proteins, including XPC and XPA. The damaged strand is then incised on both sides of the lesion by endonucleases, followed by the recruitment of DNA polymerases and ligases to fill the gap. NER is crucial for the maintenance of genomic stability and the prevention of UV-induced skin cancers. Mismatch repair, MMR, is a DNA repair mechanism that corrects errors that occur during DNA replication, as well as DNA lesions caused by DNA-damaging agents. MMR identifies and removes mismatched bases and small insertions or deletions that can lead to frameshift mutations. The recognition and strand discrimination are carried out by MMR proteins, such as MLH1, MSH2, MSH6, and PMS2. MMR ensures the fidelity of DNA replication and prevents the accumulation of mutations. Double-strand breaks, DSBs, are the most severe form of DNA damage and can occur due to ionizing radiation, reactive oxygen species, or replication fork collapse. Cells have evolved several mechanisms to repair DSBs, including non-homologous end joining, NHEJ, and homologous recombination, HR. NJ rejoins the broken DNA ends directly, albeit with the potential for small deletions or insertions. HR, on the other hand, utilizes an undamaged DNA template to accurately repair the DSB. DSB repair mechanisms are critical for maintaining genomic stability and preventing chromosomal aberrations. Oxidative damage to DNA is a common consequence of reactive oxygen species generated during normal metabolic processes or exposure to environmental factors. To counteract oxidative damage, cells employ various repair mechanisms, including base excision repair and nucleotide excision repair. These pathways act on oxidized bases and other oxidative DNA lesions, ensuring accurate repair and preventing the accumulation of oxidative damage-induced mutations. Exposure to UV radiation can cause the formation of pyrimidine dimers, leading to significant distortion in the DNA helix and potential disruption of DNA replication and transcription. UV-induced DNA lesions are primarily repaired through nucleotide excision repair, which removes and replaces the damaged DNA segment. Failure to repair UV-induced DNA damage can result in the development of skin cancers, emphasizing the significance of proper repair mechanisms. DNA crosslinks are covalent bonds that bridge both strands of the DNA helix, impeding DNA replication and transcription. Cells have evolved specialized repair pathways, such as nucleotide excision repair and homologous recombination, to address DNA crosslinks. These repair mechanisms act on the DNA structure to remove and restore the normal DNA conformation, ensuring proper DNA replication and gene expression. DNA repair is facilitated by a diverse range of enzymes that recognize, excise, and restore damaged DNA segments. These enzymes include DNA glycosylases, endonucleases, DNA polymerases, and DNA ligases, among others. Each repair pathway often involves a unique set of enzymes that collaborate to precisely repair the specific type of DNA damage encountered. Understanding the functions and mechanisms of these enzymes is crucial for unraveling the intricacies of DNA repair. Impaired DNA repair processes can lead to genomic instability, characterized by an increased susceptibility to mutations, chromosomal rearrangements, and genetic diseases. Deficiencies in DNA repair pathways, either through genetic mutations or environmental factors, can disrupt the delicate balance between DNA damage and repair, contributing to the development of cancer and other genetic disorders. Comprehensive knowledge of DNA repair mechanisms is vital for understanding the causes and potential interventions for genomic instability. Genetic diseases can arise due to inherited or acquired mutations in DNA repair genes. These mutations can impair the efficiency or fidelity of DNA repair, resulting in an increased susceptibility to DNA damage-induced diseases. Many hereditary cancer predisposition syndromes, such as xeroderma pigmentosum and Lynch syndrome, are directly linked to defects in specific DNA repair pathways. Investigating the role of DNA repair in genetic diseases provides valuable insights into disease etiology and potential therapeutic strategies. Advances in our understanding of DNA repair mechanisms have opened up new opportunities for personalized medicine approaches. The identification of mutations in DNA repair genes can help guide treatment decisions, as some DNA repair defects render tumor cells more sensitive to specific therapies, such as PARP inhibitors. Integrating knowledge of DNA repair status and genomic instability into clinical practice promises to enhance patient outcomes and enable tailored treatment strategies. In conclusion, DNA repair is a complex and intricate process essential for maintaining genomic stability and preventing the onset of genetic diseases, including cancer. The various mechanisms and pathways involved in DNA repair, such as direct repair, base excision repair, nucleotide excision repair, mismatch repair, double-strand break repair, oxidative damage repair, and DNA crosslink repair, collaborate to efficiently correct different types of DNA damage. Understanding DNA repair mechanisms is crucial for elucidating disease etiology, developing targeted therapies, and advancing personalized medicine approaches. Continued research in this field holds significant promise for improving human health and well-being.